If you had the ability to choose your child's genes, would you? This is just one of the many things that the process of CRISPR-Cas9 can do. As an AP biology student and someone who has done all the research for this speech, I feel like I am well versed enough in the subject to be able to discuss it with you. Today, we will be discussing what the process of CRISPR-Cas9 is, the purpose and potential applications, and the controversies surrounding this topic. Let's first start with the process. According to Innovative Genomics Initiative, the CRISPR-Cas9 genome editing system is the process of making specific changes to the genetic code of a cell and/or organism. This process can be broken down into 4 simple steps. Step 1. 2 different types of RNA, crRNA and transactivating RNA, bind to the Cas9 enzyme. These RNAs are also known as guide RNAs. And the Cas9 enzyme is an enzyme that is allowed to cut through DNA strands. After the binding process, we move on to step 2. This step is where the guide proteins, and also known as guide RNAs, allow this Cas9 enzyme to travel across the DNA strand until they find the exact sequence that has been programmed into the RNAs. After finding that sequence, we move on to step 3. In step 3, the guide RNAs allow the Cas9 enzyme to open up the double helix DNA strand. The Cas9 enzyme then proceeds to cut the DNA strand using 2 molecular blades. Now in step 4, repair enzymes then come and fill the gap with new genetic information. This then seals the gap and creates a new genetic code. Now that we know what the process is, we can move on to the potential applications and purposes. There are many different purposes and potential applications of this process. According to Life Science, one of them is correcting genetic diseases and sicknesses such as cystic fibrosis. As well as correcting genetic diseases, it also allows for research on human health, agriculture studies, and bioenergy. An example of agriculture studies is genetically modifying corn. For example, genetically modifying corn so it's resistant to a type of bug or a strain of pesticides. As well as that, it also allows us to choose what genes to pass on in human embryos. This is called germline editing. Now that we've discussed the process and potential applications and purposes, we can move on to the controversies surrounding this topic. As this topic becomes more widely known and discussed, the questions of morality and ethics arise. According to Dr. Rachel Ribben and Life Science, there are mistakes called off-target effects. These off-target effects are caused when instead of editing at a specific location, it edits somewhere farther down the line. This can lead to unwanted mutations. As well as unwanted mutations, Life Science also states that it can have potential negative ecological impacts. Mutations may spread from another organism to another organism and from species to species. If one species gets mutated, it can affect all the other species in the ecosystem. This can have detrimental and damaging effects. In conclusion, hopefully now that we have finished discussing the controversy, you understand what CRISPR-Cas9 is, the purposes and applications, and the controversies. And now that I've discussed all this information with you all, would you still answer the same to my beginning question? If you could choose and even change your child's genes, would you? And why? Thank you.